Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine if we could truly slow down the relentless march of a devastating disease like Parkinson's. For decades, it's been such a challenge impacting millions of people all over the world. But what if a new discovery, maybe focusing on just a tiny faulty protein in the brain, could offer a real glimmer of hope? That's exactly what we're diving into today. We're unpacking some really groundbreaking new research. This comes from the University of Sydney. It's detailed in a recent health news piece, but also, crucially, in a deep scientific paper published in Acta Neuropathological Communications. So our mission here is to explore how targeting, well, a very specific brain protein with something quite common like copper might actually pave the way for future treatments for Parkinson's. We'll get into the what they found, the how they figured it out, and maybe most importantly, the what next for this really exciting development. And just to give you a sense of the scale here, Parkinson's is the second most common neurological condition right after dementia. In Australia alone, it affects over 150,000 people. So, this isn't just abstract science, it's deeply personal for so many.
It absolutely is. And to really appreciate what's new here, we need to look back a bit at the groundwork. Professor Kay Double's team over at the University of Sydney, they've spent more than 10 years really digging into the biology behind Parkinson's. It's been a long road.
Yeah, and this is where the story gets, well, really interesting. Back in 2017, that same team made a critical discovery. They were the first to identify an abnormal form of protein. It's called superoxide dismutase 1. We'll probably just call it superoxide dismutase 1. They found this faulty version in the brains of people who had Parkinson's. Now, normally, the superoxide dismutase 1 protein is actually protective for the brain.
That's right. It's what we call a middle protein. It needs metal ions, especially copper, to work properly and sort of shield the brain.
But in Parkinson's patients, something goes wrong. This protective protein, it becomes faulty, it starts to misfold, then it clumps together, and that's when it damages brain cells, particularly those really important dopamine-producing neurons.
Exactly. The very cells whose loss drives the main symptoms of the disease. So to understand this better, and crucially to test potential fixes, the team knew they needed a good model, something that really mimics what happens in the human brain. And that led them to develop this novel mouse model. Quite an achievement, actually. They call them superoxide dismutase 1 overexpressing copper transport protein 1 knockdown mice. It's a mouthful.
Okay, so these specific mice, what was special about them?
Well, they were bred very carefully to have two key features. First, high levels of the human superoxide dismutase 1 protein, and second, reduced levels of copper, specifically in their neurons. And these are precisely the kinds of changes you see in postmortem brains from Parkinson's patients.
Ah, okay, so it mirrors the human condition.
It does. And as a result, these mice, they developed significant problems linked to that human superoxide dismutase 1 protein. They showed motor impairments that got worse with age, trouble with movement, and critically, they lost a significant number of dopamine neurons in that key brain area, the Substantia nigra. So this gave the researchers a living system to really test ideas.
Right. So you've got this model that acts like Parkinson's. Now you can test treatments. And this latest study, it builds right on that earlier work, focusing on that faulty superoxide dismutase 1 protein. So how did they try to fix it? What was the approach?
They use a special copper supplement. It's a drug called diacetylbis-4 methylcellsemicarbazonate of copper 2.
Oh, okay. Let's maybe call it this unique copper compound.
Yes, that's probably wise. So this unique copper compound is key because it can do something many drugs can't. It can cross the blood-brain barrier. It actually gets into the brain where it's needed.
Which is a huge hurdle for neurological treatments, right? So many things just don't get past that barrier.
Precisely. A major challenge to overcome there. So for the study design, they took these specialized mice, the ones already showing Parkinson's symptoms, and divided them into two groups. Carefully done. One group got a daily dose of this unique copper compound orally for 3 months. A specific dose, 15 milligrams per kilogram. The other group just got a placebo, a dummy treatment.
Okay, makes sense. And what were they measuring? What were they looking for?
They looked at several things very comprehensively. Motor function, obviously how well the mice could move, their coordination. They measured the volume of that faulty superoxide dismutase 1 protein building up in brain regions like the Substantia nigra. They also looked closely at the protein itself, its chemical structure, what we call post-translational modifications, to see how it was changed. And they measured its enzymatic activity, basically, how well the protein was doing its job. Finally, they counted the surviving dopamine neurons and measured dopamine levels in another brain area, the striatum, which is also vital for movement control.
Wow, okay, so behavioral and cellular level detail. And the results. You mentioned they were remarkable.
They really were. Professor Double herself said all the mice we treated saw a dramatic improvement in their motor skills, which, as she said, is a really promising sign.
A dramatic improvement. So the placebo group declined, as expected.
Yes, the placebo group showed worsening motor symptoms over time. But the mice getting this unique copper compound, they essentially didn't develop those movement problems.
That's incredible.
It is, and they quantified it. On a balanced beam test, for example, the treated mice were much better. They crossed faster, they slipped way less often.
Yeah.
They performed almost like healthy mice, mice without any Parkinson's issues.
So not just slowing it down, but preventing the decline and maybe even restoring function.
The data point strongly towards restoration, yes, a striking rescue of motor function. And it wasn't just behavioral. At the molecular level, the effects were just as profound. This copper compound treatment led to a really significant drop, a 4.4-fold decrease in the amount of those faulty superoxide dismutase 1 protein clumps in the Substantia nigra.
4.4-fold, that's substantial.
It is. And importantly, this reduction was directly linked to the copper levels in that part of the brain. More copper meant less faulty protein, a clear connection.
Okay, but you mentioned something surprising earlier about astrocytes, the support cells.
Ah, yes, that was fascinating. Astrocytes, these star-shaped glial cells, they do a lot of support work, including managing metals like copper. What the study found was that this faulty superoxide dismutase 1 protein was actually accumulating more within the astrocytes than inside the dopamine neurons themselves, at least in the main cell body.
Really? So not primarily in the neurons we always focus on.
It seems the astrocytes are a major site of this specific pathology in this model. And here's the kicker. The copper treatment was incredibly effective there. It caused an 8.5-fold decrease in the faulty protein within astrocytes.
8.5, that's huge.
Much bigger than the effects seen directly in the dopamine neuron cell bodies, which was more like a 2-fold decrease. So this really highlights astrocytes as potentially a key player and a primary target for this kind of therapy. It slightly shifts our view.
Definitely changes the picture. And did the treatments just clear out the bad protein, or did it fix the protein itself?
That's another crucial part. It did largely restore the protein's normal function. They looked at those chemical modifications we mentioned earlier, and the treatment corrected 29 out of 40 of them. That's 73%, bringing them back towards normal levels.
So it fixed the chemical glitches.
Yes, things like oxidation on key parts of the protein, changes in glycosylation, things known to make it misfold and clump. These were reversed. And perhaps just as importantly, the treatment significantly boosted the protein's enzymatic activity, getting it back towards healthy functional levels. And again, this functional improvement was strongly tied to the amount of copper present in the tissue.
So it's not just removal, it's actual repair and functional restoration of the protein.
Precisely, making it work properly again. And all of this, this molecular and cellular repair, it had a direct impact on the dopamine system, the core issue in Parkinson's. As expected, the untreated specialized mice had low dopamine levels, high dopamine turnover, and significant neuron loss in the Substantia nigra.
Right, the classic Parkinson's signature.
But the mice treated with this unique copper compound. The treatment prevented that loss of dopamine neurons. It also rescued dopamine levels and turnover in the striatum, keeping them close to the levels seen in healthy mice.
Wow, so it protected the neurons.
It protected the neurons and restored the chemical balance. And again, it all correlated. Higher neuron survival linked to higher copper levels, lower faulty protein load, and better superoxide dismutase 1 activity. It ties everything together very neatly.
Okay, so connecting all these dots, what's the big picture implication here for humans with Parkinson's? Professor Double suggested this could potentially slow the disease progression in humans, right? After more studies, of course.
That's the hope, yes. This study provides the first real evidence in a living system that giving copper in this specific form can fight back against this particular type of protein pathology found in Parkinson's, and that it can improve the survival of those critical dopamine neurons. It identifies a potential new therapeutic pathway.
A treatable pathway, potentially, for many Parkinson's patients.
It is a very promising pathway. But, we have to be realistic about Parkinson's. It's complex. This faulty superoxide dismutase 1 protein, it's likely just one piece of a much larger puzzle.
Right, not the only thing going wrong.
Exactly.
Yeah.
Think about treatments for other complex diseases, like HIV. It's usually a cocktail approach, right?
Multiple drugs targeting different things.
Parkinson's might be similar. This copper approach could be one important component. On its own, maybe a modest effect, but combined with other therapies targeting different aspects of the disease, that could lead to really significant overall benefits.
Makes sense. Adding tools to the toolbox. Now, this is all incredibly exciting, but we should also talk about the limitations and the next steps. How easily does this translate from mice to people? That's always the big question, isn't it?
It absolutely is, and there are important points to consider. First, the mouse model involved genetic changes that were present everywhere in the mouse's body, globally. In human Parkinson's, the faulty protein seems more concentrated in specific brain areas that are degenerating. It's not necessarily brain-wide from the start.
Okay, so the model isn't a perfect replica of the human regional pattern.
Correct. And also, the really strong motor improvements in the mice might be because the treatment was helping in multiple brain regions affected by the faulty protein, not just the Substantia nigra, which is the main focus in humans.
Ah, so the effect might be more localized, potentially less dramatic in humans.
It's a possibility we need to consider. It might mean we need even more targeted delivery methods down the line.
And what about the copper itself? You mentioned it needs careful management.
Yes, that's critical. The copper deficiency seen in Parkinson's isn't usually brain-wide either. It's often specific to those vulnerable regions. And interestingly, the study found that if you gave this copper compound to mice that had normal copper levels already, it could actually lower their dopamine levels in the striatum.
Oh, so too much or giving it where it's not needed could potentially be unhelpful or even harmful.
Exactly. It's a very fine balance. You need to boost copper where it's low, but avoid overloading areas where it's already sufficient.
Which leads to a big practical problem.
Which is that right now we don't have a good, reliable way to measure copper levels accurately inside different regions of a living human brain.
Right. Without that, personalized dosing is really difficult. A major hurdle.
A major hurdle for clinical application, yes. And there's another subtlety. The treatment, even though it was very effective, didn't completely eliminate all the faulty protein pathology.
Okay, so some remained.
A small amount, yes, which suggests that maybe some of this protein problem happens through ways that don't involve copper directly, copper-independent mechanisms,
Implying
that maybe combination therapies will be needed even for this specific protein issue, targeting those other mechanisms too.
Or perhaps different approaches altogether.
Possibly. You could think about therapies that just reduce the amount of superoxide dismutase 1 protein being made in the first place. Things like antisense oligonucleotides, which can kind of switch off specific genes.
Genetic therapies, okay.
But that has its own challenges. Superoxide dismutase 1 is an important antioxidant. Reducing it too much might have unintended negative consequences, potentially making neurons more vulnerable in other ways.
Right, it's a protector normally, so reducing it is tricky.
Very tricky. It's always a balance in biology.
So wrapping this up then, this deep dive, it really shows compelling evidence from this University of Sydney research. A specific form of copper supplementation can fight this faulty superoxide dismutase 1 protein problem, protect dopamine neurons, at least in this model system. It really does open up a novel, potentially treatable pathway for a lot of people with Parkinson's, a new direction.
It absolutely does. And think about the ripple effects, too. Beyond just treatment, what if this spurs the development of better diagnostic tools? Imagine if we could accurately measure brain copper levels non-invasively in people.
That would be a game changer for identifying who might benefit most from this kind of copper therapy.
Precisely. Making it truly targeted. It just underscores, the incredible complexity, but also the really exciting future we're looking at in neurological research.
So here's something to think about as you go about your day. Given these findings, especially about the regional differences in copper and the likely need for combined treatments, how might future therapies for Parkinson's become incredibly personalized, really tailored down to an individual's specific biological profile? What steps are needed to make that a reality? This research feels like a really significant step on that journey.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.